But if you notice, you saw differentiation of T cells from a CD8 to a CTL. You saw how CD8 kills cells. You saw trafficking, because he mentioned CCL21. You saw signal 1 and signal 2. And you saw T cell development, because he mentioned double positives. So like a lot of the stuff we've been talking about all show up there. So that's that. Plus some, tabloid fodder. All right, so now we're going to get back into thinking about the specifics of T cell effector function. So if you recall, we left off last time talking about helper T cells and this idea that we have a naive CD4 T cell, sometimes known as a Th0 cell, that will interact with antigen on an antigen-presenting cell and will receive some kind of cytokine. And so the cytokine it receives is sort of on these top arrows. That will turn on a particular transcription factor and make that T cell stop being a naive Th0 and start being a T cell of a particular subtype. And one of the things that will do is it will make a characteristic effector cytokine. Each different one of these cell types is really important for dealing with a different type of microbe. And if it goes wrong, it can be associated with a different type of disease. So last time we saw Th1 and Th2s, which were sort of the originals that were described. Th1s are differentiated by IL-12. They make TBET. And then that allows them to transcribe interferon gamma. So they combat intracellular pathogens by activating macrophages and CD8s. Th2s get IL-4. This tells them to make this transcription factor GATA3. GATA3 allows them to transcribe IL-4, and then they can turn on eosinophils and mast cells to combat extracellular parasites. We talked about Th17. Th17s receive an inflammatory cytokine like IL-6 along with TGF-beta. This makes them turn on the transcription factor ROR-gamma-T. ROR-gamma-T tells them to make IL-17. This is really useful in turning on neutrophils to combat extracellular pathogens. And we talked about T follicular helpers, which are polarized by a combination of IL-6 and IL-21 to make the transcription factor BCL6, and then make these effector cytokines IL-4 and IL-21. And this is going to be really important for turning on B cells in the follicle. FH is for follicular helper. So this is how B cells are going to get turned on in the lymph nodes. So for the second part of what we're talking about today, the T follicular helper will be really useful. However, we kind of didn't really get to talk fully about the final one of these T cell subsets, which are the regulatory T cells. In reality, regulatory T cells were actually the third of these types described. There was a short period of time where they were called Th3s. They kind of require a little bit of talking about on their own, so I want to make sure that we kind of get the details of regulatory T cells. So that's where I left us off, and so let me move specifically to the regulatory T cell info. Okay, so regulatory T cells are polarized by TGF-beta. So TGF-beta is their polarizing cytokine. And notice that this is sort of a slight similarity with Th17. Th17s also included TGF-beta for polarization, but with Th17s, it was TGF-beta plus an inflammatory cytokine, like IL-6. Here, it's TGF-beta alone, so there's no inflammation in this case. Once the cell receives TGF-beta, it starts making the characteristic transcription factor, FOXP3. In fact, FOXP3 expression is one of the big ways we actually figure out if a cell is a Treg. There's a lot of FOXP3 sort of experiments people do. So the transcription factor is FOXP3. And then those regulatory T cells make this cytokine called IL-10, which is the effector cytokine. The general function of regulatory T cells is to inhibit the activation or turn off other cell types. Once upon a time in the 80s, people thought they had found a type of cell called a suppressor T cell. They figured out some genetics about the suppressor T cell. Suppressor T cells were like the cool hot thing. Then they actually sequenced the genome, and they realized the gene that was the suppressor T cell gene didn't exist because they had messed up somewhere in the experiments. And then it was suddenly like overnight, all immunologists were like, I don't know what you're talking about. I never believed in suppressor T cells. Suppressor T cells aren't a thing. And like the word suppressor still really has like is not used by immunologists in the field. Like we just don't use suppressor. And so it actually weirds me out that they talk about suppression of cells even on this figure. Because like we so never use the word suppressor and suppression. We often use regulation, which is our nice way of saying inhibit or suppress. So Tregs have this ability to turn off other immune responses. So they can turn off inflammation, they can turn off other T cells, they can turn off basically all of the different T cell types. And so they're really important, you can imagine, for helping deal with autoimmunity and things like that, because they're going to be turning everybody else's responses off. And if you didn't have them, that would be a bit of a big problem. So this is sort of, I think, a relatively easy way of understanding regulatory T cells. Here you can see a Treg actually inhibiting another CD4 T cell. But one other thing that is important to know about regulatory T cells is gravity. But also that there are actually two ways a cell can become a regulatory T cell. And so what I'm showing you here on this slide is only one of the two ways a cell can become a regulatory T cell. It turns out you already know the other way, you just don't realize you know it yet. The other way that a cell can become a regulatory T cell is actually that some T cells leave the thymus as regulatory T cells. So they don't actually get polarized and start differentiating in the periphery, which is what I was just showing you on the previous slide. These cells are actually told you are going to be a regulatory T cell in the thymus, and leave the thymus already committed to being a regulatory T cell. And so if you remember when we talked about T cells developing in the thymus, we talked about cells that had no signal. So we can even say no or low signal. If you remember, those cells underwent death by neglect. We had sort of this low intermediate. Those cells were positively selected. And they become a T cell. And we had cells that got a high signal. Those cells were negatively selected. And died. And I said, well, yeah, there's a fourth thing that happens. And I just sort of waved my hand at it. That fourth thing is if it's still kind of a middle signal, it's still an intermediate signal, but it's on the higher side. So if we have cells that are getting sort of this, here they're calling it intermediate and they're calling this one low. You can sort of think of it, and as long as it's clear what we're calling these four things, you can call them different things, whatever. That cell gets a high intermediate signal. It's not quite high enough that you think it's dangerous and you want to kill it. But you're also not totally sure you want it to go into the periphery and be ready to attack stuff. That cell actually becomes a Treg. That cell is pushed to start making FOXP3 while it's in the thymus. And so you could turn on FOXP3 because you receive TGF-beta, or a cell could turn on FOXP3 due to the signals it got in the thymus. And that will also make that cell be a regulatory T cell. So this cell might be a little more risky at being self-reactive, but in fact, if it sees its self-antigen, it's going to turn things off. So it's sort of a nice safety mechanism. So Tregs can come in from two places, not just the one that we were seeing before. And in fact, this is the real version of the figure from your textbook. When I showed it to you before, I put white boxes over some things, which was this idea of cells developing to become Tregs. And so you can see that there is this fourth option where some of our cells are going to develop to be Tregs. And so we will have some CD4s come out of the thymus. Those regulatory T cells that make FOXP3 are able to inhibit other types of responses in a number of different ways. They can do that by making cytokines like TGF-beta as well as IL-10, which can directly inhibit other cells. They can sometimes be cytotoxic, though I'm not going to dwell on that. They can actually make some cytokines that can turn off antigen-presenting cells and stop antigen presentation. And they have a ton of IL-2 receptor. It's actually part of their initial discovery was the details of the IL-2 receptor. And they can also basically just steal all the IL-2 so that other T cells are deprived of IL-2. And so sometimes this is called being an IL-2 sink. And so sometimes they also just take all the IL-2 so there's no IL-2 for other cells. And so these are some of the ways that there can be inhibition by a regulatory T cell. There are also situations where regulatory T cells can inhibit by cell surface proteins and contacting other cells. So instead of secreting something like TGF-beta or IL-10, they can actually also have a transmembrane protein that suppresses, though that is not shown here. And so the other kind of piece to all of this to mention is that when we initially described the Th1, Th2 phenomena, one part of that, if you remember, was feedback between the Th1 and Th2 cells. So once you started making a Th1 response, you got more Th1 kinds of cytokines, and you pushed any other cells that hadn't decided yet if they were going to be Th1s or Th2s, you pushed them towards Th1, and you also inhibited Th2 generation. So you were kind of pushing the whole response towards Th1 and inhibiting Th2. With Th2, you were doing the same thing. You were pushing towards Th2, inhibiting Th1. Of course, now we know that there are way more subsets than just Th1 and Th2. And what we also realize is that they all feedback on each other. And so each type of T cell inhibits the generation of the other and has positive feedback on its own subtype. So you can see Th2s inhibit a Th1. They also inhibit a Th17. Th1s inhibit a Th17 or a Th2. Tregs inhibit everybody. So basically, we could make some sort of very complicated web diagram where every type of T cell inhibits, or every type of CD4 T cell subset inhibits the generation of the other CD4 T cell subsets, while also giving positive feedback on its own subset. The next two slides are slides I had put together for this sort of set for Monday with the specific idea that they were helpful review summaries for you when you were studying for the exam. So just so you know, here is a table from your textbook with all of the information about helper T cell subsets. And here is a figure from your textbook with all of the information about helper T cell subsets. And so in theory, that is where I would have ended on Monday if I didn't have to leave early. And so now I'm going to switch to what I wanted to talk about today. And that topic is B cell responses in the periphery. So we just talked about T cell responses in the periphery and how T cells act in the periphery, but now we're going to kind of deal with the same questions with B cells. So when we last saw our B cell, in the last episode of What B Cells Do, our B cell was undergoing B cell development in the bone marrow and sort of heading out to go to the periphery. And so we're going to see some actions in the periphery today of those B cells. So now we're in the periphery. We are seeing this B cell could see antigen and actually do something about it, which will include making large amounts of antibodies. This process is happening in a secondary lymphoid organ, like a lymph node, just like all the events we've just been seeing with T cells. And so the trafficking pieces of this are actually almost identical to the trafficking pieces we've already seen. So I don't need to tell you about trafficking. So we're going to, and in fact, our secondary lymphoid organs are going to include antigen coming in through the lymph, as we talked about before. We're going to have antigen-presenting cells, as we talked about before. We're going to have T cells, and we're going to have B cells, all in those secondary lymphoid organs. And all of these things are going to be important in this process. B cell biologists would probably yell at me for reducing this information to only one slide, but I am going to reduce this information to only one slide, which is that there are multiple ways to turn on B cells in the periphery. Some of them involve T cells. Some of them do not involve T cells. The ones that involve T cells are called T-dependent responses, or and antigens that are usually go through this kind of response are called T-dependent antigens. There are other antigens that are T-independent, and so B cells can be activated in T-dependent or T-independent ways. For a B cell if a B cell is activated in a T-dependent way, it is going to actually get some cytokines and some protein-protein interactions from a T cell. If the B cell is getting activated in a T-independent way, it is still getting multiple signals, but those signals will come through other means. They might be through the B cell receptor and through something like a TLR, some kind of pattern recognition receptor. They also can sometimes be through the B cell receptor and some other protein. This one is showing us actually a complement receptor. So you've got this complement receptor binding to complement that's bound to the antigen, and we're getting some signals for the complement receptor and through the B cell receptor. And so you can see this idea of either T-dependent or T-independent antigens. Pretty much everything I say, other than this slide, is going to be about a T-dependent response. A T-dependent response involves things like have class switch happening. So yes, class switch. It involves us having affinity maturation, which we talked about previously. It involves us getting memory cells. And it really can involve particularly large amounts of antibodies as well. T-independence absolutely happen. However, they don't have these same awesome qualities of a T-dependent response. So we're not going to get class switch. We're only going to be stuck with IgM. We're not going to get affinity maturation and get that high binding. We're not going to get a good memory response. So these happen, but we're going to largely talk about a T-dependent response that has all of these features. One other thing is very important about a T-dependent versus T-independent antigen. Only one kind of biological molecule can be a T-dependent antigen, and that is a protein. So if you recall when we talked about what could be an antigen for a B cell, we said proteins, or nucleic acids, or lipids, or carbohydrates can bind to B cells and bind to antibodies. If we have a polysaccharide, if we have a lipid, if we have a nucleic acid, it can be a T-independent antigen. It cannot be a T-dependent. Only proteins can be T-dependent antigens. And the reason for that, this cannot be stressed enough, honestly, is because a T-dependent response shockingly involves T cells. And T cells only recognize proteins. And so if you want to get a T cell involved, there has to be a protein antigen, because T cells only see proteins. If you have some other type of biomolecule as your antigen, you can turn the B cell on, but you're not going to get the nice T-dependent response. And so everything else I'm going to tell you is about T-dependent responses. But you should know that T-independents do exist. And this just shows you the sort of excellent outcomes of what will happen to our B cell if it is activated in the periphery along with a T cell in a T-dependent response. So if we have our B cell and T cell interact, that B cell will proliferate, hooray! It will differentiate, just like we saw our T cell differentiate. So it's same general things. That B cell will potentially class switch, as well as undergo somatic hypermutation. So all of those things are going to be happening in this B cell. We can think about the same questions we thought about with the T cell response in terms of, why does it take so long? Why is it that after I get my vaccine, I am not immune in 4 minutes? Why does it take like a week? What the heck is going on? And the first answer is trafficking, just like it was for T cells. We have to wait for the antigen to get into the secondary lymphoid organ. And the B cell and the antigen have to interact. But then the B cell and the T cell have to interact. And in fact, that T cell also had to go through all this business before we can actually have this interaction do anything. So we also have to have all the T cell stuff happen before we can make this work. That's why I had to tell you about the T cell stuff before I could tell you this. And then the B cells actually start to differentiate and do other things afterwards. So we're actually dependent on all of those T cell responses that we have. So the first part of this, you can see, does involve the B cell interacting with the antigen. What that implies is that the antigen is in the secondary lymphoid organ, along with the B cell. The B cell gets there through trafficking, like we saw before. The antigen, this is slightly weird. I'm going to do this slightly differently. I don't like this slide here. I like this slide here. The antigen is actually going to come into the lymph node via the lymphatics. So remember, we talked about how the lymphatics will just have antigen flowing in, getting collected in the liquid throughout the body. And that antigen will get into the lymph node. We have a cell type in the lymph node that is going to be really important here. This cell type is called a follicular dendritic cell, or an FDC. FDCs are everyone's favorite. And by favorite, I put that in air quotes. Most important thing you should know about a follicular dendritic cell is that a follicular dendritic cell is not a dendritic cell. It got named a dendritic cell because it looked like one under a microscope. It had the same kind of stretchy, projection-y structure. But then when scientists studied them in more detail, they realized they're not dendritic cells at all. So first of all, their name is confusing. What that means is they do not do phagocytosis. They do not take antigens into them. We don't think about them presenting antigens on MHC, especially class 2. They're nucleated cells, so sure, they present on class 1. But they only live deep in your lymph node. They're probably not getting like infected super often. So MHC is not the main thing that we think about when we think about follicular dendritic cells. Instead, the thing that we think about with follicular dendritic cells is that follicular dendritic cells are covered with any type of receptor you can imagine. So they are really, I like to think of them as being really, really, really sticky. Here is another example of a follicular dendritic cell. You can see the follicular dendritic cell. It has that kind of structure, so it has lots of surface area on its surface. And it has all sorts of receptors. It might have a complement receptor. So here, see, you can see complement receptor here. It might have Fc receptors. It just has so many receptors that it sticks to everything. I like to think of a follicular dendritic cell as a piece of fly paper. You ever seen those paper? Sometimes people hang up and it's just sticky paper and flies fly by it and they get stuck to it. And then you don't have flies anymore. That's basically what I think of a follicular dendritic cell as being. Is just this nice sticky cell in the lymph node. So any antigens that come into the lymph node get stuck there. And so in some ways I think about it as sort of like an antigen repository. It's just like a bank of antigens because all the antigens are stuck all over it. And so you've got this nice antigen bank cell. And so that can be really important for that B cell to encounter antigen. That B cell will get activated by that antigen. So the B cell will go from being a resting B cell to an activated B cell. And it could see that antigen as free antigen, might float on by, may also see it on an FDC. So our B cell is going to encounter antigen, and that's going to activate our B cell. The signaling cascade looks incredibly similar to the one we saw for T cells. And as a result, that B cell is going to start doing things like making more MHC, making more B cell receptor, and also making B7. Remember, that can interact with T cells. That B cell, when it binds antigen on its, using its B cell receptor, will also internalize that antigen. And so the B cell will be activated, will have all these new proteins on its surface, and it will also bring in antigen using its B cell receptor. So that antigen will now be in an endosome because it has become endocytosed by that B cell. If you think about it, an antigen that is endocytosed by a B cell could get presented on MHC class 2 because B cells are one of our types of professional antigen presenting cells. And so B cells will internalize that antigen, but oh my gosh, now we put it in the class 2 space. And in fact, that antigen, if it's a protein, will get degraded, will go on MHC class 2, and the B cell will actually start presenting the antigen that activated it on MHC class 2. So you can see all of this happening here. You don't see our B cell yet secreting antibody or doing any of that good stuff. Because in order for our B cell to actually do any of that good stuff, this B cell still needs to get help from a CD4 positive T cell. Realize our B cell is now presenting antigen on class 2. It's basically saying, hello, T-cell, help me please. T-cell, I got this antigen. Something bad is happening, please help me, right? And our T-cell is going to be a CD4 T-cell, so it can recognize MHC class 2 plus peptide and interact with that B-cell. And that T-cell is going to give that B-cell some signals that will allow the B-cell to further be activated. When we talked about T-cell activation, we talked about this idea that T-cells could get 3 signals. There was a signal 1, a signal 2, and a signal 3 for the T-cells. What were the 3 signals for the T-cells? Yeah, Jen. So signal 2 was co-stimulation. It was B7 on the antigen presenting cell, CD20 on the T cell. Yeah, so that was signal 2. What else? What about signal 1 and signal 3? Yep, Sebastian. So yes, but what was the receptor for? Cytokine. So signal 3 is some kind of cytokine. And then there's signal 1. Yeah.
Signal 1 was transcribing IL-2.
Yep, through the, what kind of receptor? On a T cell. Aaron. TCR. The T cell receptor is signal 1. Signal 2 was co-stim. Signal 3 was cytokines. For our B cell to fully get activated, it needs 3 signals. The first signal is the signal we just saw from the antigen through the B cell receptor. It's signal 1. So with T cells, signal 1 is the T cell receptor. With B cells, signal 1 is the B cell receptor. So very similar. The B cell then needs to get signal 2. That signal 2 in this case is coming from the T cell. So it's a protein-protein interaction between the T cell and the B cell. So the T cell is sort of brought close in this interaction in order, based on MHC class 2 and peptide. And when the T cell is brought close, that allows the T cell to provide this protein for the interaction to give the B cell signal 2. This includes a T cell making this protein called CD40 ligand, which interacts with a protein called CD40. And if you actually listen closely to that Ben and Jen thing we heard earlier, the friend, the CD4 cell, was making CD40 ligand. So this is giving our B cell signal 2. The T cell also makes some cytokines in order to give the B cell signal 3. So again, conceptually, it's the same 3 signals. And when that B cell gets those 3 signals, it is fully activated and can go into some of these other processes. I told you about sort of the other receptors involved in T-independent responses. You can kind of think of those other receptors as alternative signal 2s. They're just not as good as this signal 2. So we can also kind of think a little bit about details and where this happens. So to start with, I know this is shocking. Our B cell enters into the lymph node, and the place the B cell goes is called the B cell zone. So the B cell will go to the B cell zone, and it will see antigen. The B cell will then move. And the reason why it moves is it wants to find a T cell. And so that B cell is going to move towards the T cell zone. The B cell is in fact going to kind of move to the edge of the B cell and T cell zone. And once the B cell does that, it is going to move to another location. This location is called the germinal center. And the germinal center is very key for B cells to be able to make their best possible responses. So if you want an awesome B cell response, you need to have that B cell go through this germinal center process. Germinal centers are slightly weird to define because technically a germinal center is where the germinal center response is happening. And you're like, yeah, thanks, Captain Obvious. It's in, as you can see, this sort of outer area of the region. And the one thing that is key about a germinal center is that germinal centers form around follicular dendritic cells. So you can sort of imagine, you could say the B cell now goes to a germinal center. Or you could kind of also say the B cell now goes to an FDC and builds a germinal center there. Like, which of those is the correct phrasing is a little tricky. This is all in the follicle of the lymph node. And there are some cells there, T follicular helpers, that live in the follicle, that are going to help the B cell through this germinal center process. And so I already told you about follicular dendritic cells. And now I can tell you about the germinal center and the germinal center reaction. So here you can see a germinal center. They are easily distinguishable in lymph nodes. So if I were looking at, if I took one of your lymph nodes and looked at it under a microscope, especially if you knew you were sick and had an active infection, I would find germinal centers easily, and I could identify them under the microscope super easily. On Friday, we're going to be talking about a paper in here. That paper involves a lot of discussion of germinal centers, and it involves some of the prettiest images of germinal centers I've ever seen in my life. That's one of the reasons why I like this paper so much, is because it has germinal center images, and one of its big points is about the germinal center, and the images are kind of obvious. So we're going to see germinal centers more on Friday. The germinal center has a few different areas. So the germinal center has an area called a dark zone and a light zone, which you can see here. And we can first look at the dark zone and the light zone in this image on the right. So if you look at the germinal center and the dark zone and the light zone, you can see there's darker staining in the dark zone and lighter staining in the light zone. If you look at the sort of darkness versus lightness in those two zones, why might you think, why is there such a big difference in darkness versus lightness in those? Yeah, Jamie? Density of cells. The dark zone has way more cells. That makes it darker, harder for you to see what's going on there. The light zone has way fewer cells. That makes it easier for light to go through, easier to see. And in fact, that's actually something you can think about in terms of what's going on there, because the dark zone is the place where you have lots of cell division. And so it's crowded. And the light zone doesn't have a lot of cell division. In fact, you might imagine it has a bunch of cell death. And so it's not as crowded. The cells are going to start their time in the germinal center in the dark zone. So remember, the first thing that's going to happen here is our cell is going to get this signal from the T cell. It's going to get signal 1, signal 2, signal 3. It's pat on the head. And we're going to say to that cell, you're good, you need to be activated. You're going to differentiate. Remember that our T cell, the first thing it did before it differentiated was divide. First thing our B cell is going to start to do is divide. You can see our B cell is going to proliferate a lot, make more copies of itself, because now it's useful. We want to have more of them. And so that cell is going to come in to the germinal center, and it's going to divide. And as a result, it's going to make lots of progeny. I want you to imagine like it makes like 1,000 progeny. And there's so many, and there's so many of them that it gets crowded. So that's the dark zone. But there's some other things that are going to happen in the germinal center. So you can see our cell starts out, it interacts with a T cell, it gets activated through T-dependent activation, and goes into this germinal center where it starts to proliferate. Here it shows it makes 2 progeny. Really, we'll say it makes 1,000. It also is going to do another process here. And that process is a process of somatic hypermutation. So this B cell is going to turn on an enzyme that gives it mutations in its DNA. It's a somatic cell, it's a body cell, and it's a lot of mutation. So it's somatic hypermutation, SHM. So that cell is going to start mutating. Now remember that we made 1,000 progeny. Each of those 1,000 progeny is going to start mutating. And you can imagine each of those 1,000 progeny might make slightly different mutations. So we're going to take this cell that was a pretty good cell. It got a signal, got a pat on the head. And we just made 1,000 copies of it and tweaked all of them a little bit. So we made 1,000 copies of that cell, each they're slightly tweaked. Some of them are going to end up with a receptor that's even better than the original because of those mutations. Some of them are going to end up with a receptor that's the same as the original. Some of them are going to end up with a receptor that's worse than the original. Those cells are now going to move to the follicular dendritic cell and the T cells and see if they can still see antigen. If mutation destroyed the B cell receptor, then we're not going to see antigen, and the cell's going to die. No pat on the head signal. If the cell binds antigen really well, it's going to see antigen here, and it's going to live. And in fact, you can kind of think of this as a competition. In fact, this is survival of the fittest in your lymph node with cells. So there's going to be a competition. Of those 1,000 progenies, the ones that bind antigen the best are going to win and get the best signals and sort of crowd out anybody who's weaker than them. And so this process is going to improve the response of the B cells. And the reason why it's going to improve is both because you get the mutations, but then you select only the cells that made good mutations. The ones that made bad mutations die. They don't get a signal anymore. So our B cell is going to hopefully make some mutations. The enzyme that makes these mutations specifically just mutates the variable regions of the heavy and the light chain so that the antigen binding region of the antibody changes. So you can see that we get this better binding here. The change to the DNA is called somatic hypermutation. That alters the antibody to have better affinity, and so the affinity matures. So we call it affinity maturation. This is actually something that immunologists have been talking about a lot because this is sort of this idea of what happens as your immune response goes on for a while. People are like, but I get a vaccine and then my vaccine goes away and then there's a variant and then what am I going to do? Your immune system's taking care of it. Because over time, your immune response is improving. We actually see better binding, and we actually see better breadth of binding. So you can actually bind to more variants. And there was a beautiful study that came out in the past year that showed if you just let your vaccine cells that come from a vaccine take some time to go through this process, they can neutralize every variant that's out there. It's beautiful. I considered having us read that paper on Friday and not the one that we're reading, but I love this paper too. I love this paper for Friday too. They were both great for this discussion. And so, in fact, somatic hypermutation, and in fact, getting a third booster dose pushes this to happen even more. So you get these even beautifuler antibodies. And so what's going to happen is that we're going to have our cell pick up these mutations. Here's our cell. It gets activated. It starts mutating. And now it's going to go to that follicular dendritic cell and that follicular helper T cell and test. Can I still interact with my T cell and an antigen? If yes, we're going to divide some more. Note, we turn off the mutate enzyme before we divide, because we don't want to have our mutator on while we're dividing. That would be bad. We actually go back to the dark zone to do the dividing. If no interaction, the cell dies by apoptosis. And in fact, the cell can go through this process multiple times. So it will go through, it will mutate, it'll test. If it goes well, it'll go back to the dark zone and divide some more, mutate some more, try again. And so you get multiple rounds of improvement in the germinal center to eventually get you to a really fantastic antibody and a really fantastic B cell. Sometimes here students ask the question, how does the B cell know when it's time to stop going through this response? And the answer is we don't know. But that's such a common question, I felt like I should address it. Eventually, however, that B cell will leave the germinal center reaction. And that B cell will now have very high affinity. It will be mutated. It will be awesome. And that B cell will now be able to differentiate. And so instead of being just a B cell, it will now have the ability to either be a memory B cell to live for a very long time, to protect you much later, or it will be what's known as a plasma cell. And plasma cells make extreme amount of antibodies. They actually differentiate so fully that they basically lose a lot of parts of the cell, and they are basically just ER and the ability to secrete antibodies. They like totally change in shape. They totally change their cell. They become highly specialized antibody production factories. And so they make high levels of antibody. We're going to have this nice maturation, so the antibody has higher binding, or some of the B cells are going to become memory B cells. We also have one more event that happens in the germinal center, which is that during this process, while the cells are mutate, 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 test, 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 mutate, 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 they also have the opportunity to class switch. And there's debate in the field about, do they class switch and then mutate, or do they mutate and then class switch? You don't care. They mutate, or they class switch sometime when they're in the germinal center. And so we also can get switching to other types of isotypes of antibody. So if you recall with class switching, our VDJ region is very close to the constant region gene that encodes M. The others are further downstream. That will give us either IgM, IgG, IgD, IgA, or IgE. This happens through the process of class switching. So upstream of each of these constant regions, there is something called a switch site, or S. This is very not to scale. This makes it look like the M and this S are like the same size. They're so not. S is like really short and M is really big, but whatever. So you can see we have this switch site before each constant region. What will happen is we will have the switch sites, when we're going to do class switch recombination, we'll actually have the switch sites recombine. So you can see there's this switch before mu, there's a switch before gamma 1. We'll actually cut the DNA again between those switch sites. So you can see we kind of make this new little switch site. We ditch all the intervening DNA, and now we just put the IgG constant region here. And so this is another recombination process. We are breaking the DNA, but it's happening in the periphery, as is the somatic hypermutation. All of these are happening in a secondary lymphoid organ in the periphery. You might wonder how it is that the B cell knows which isotype to make. Why does it decide? I would like to make IgE today. I would like to make IgG today. Whatever. And the reason why it makes that decision is based on which cytokine, which signal 3 cytokine the B cell gets. So the T cell is going to give a cytokine. And when the T cell gives a cytokine, that tells the B cell which class switch it should do. And you will see these cytokines are sort of similar to ones from different helper T cell subsets. So if we get different helper T cell subsets, we say we're going to push down this IL-4, Th2 route, then perhaps we will get some IgE or some IgG1. If we've pushed the T cell response down a different track, we get a different type of antibodies out. So this is going to really control our class switch. There is a really interesting disease called hyper IgM syndrome, where some patients do not class switch. Here you can see a lymph node just under a microscope, not really much staining or anything. And you can see how obvious the germinal centers are, right? There are some patients who are missing certain proteins who don't make germinal centers. This is what their lymph nodes look like. And this is a genetic defect. The paper that we're going to see on Friday is a different reason why germinal centers are messed up. It's not a genetic defect. You aren't born with it. This you're born with. These patients have no germinal centers. As a result, they never can do class switch. And so if you look at their antibodies, all of their antibodies are IgM. They have way too much IgM and not enough of anything else. So they have hyper IgM. They have no IgG, no IgA, no IgE, because they never can class switch. There are 3 proteins that can cause hyper IgM syndrome, 3 different mutations. One of those proteins is CD40. Where's a good CD40 image? Hello? Who thought this was a good idea for good organization for these slides? So one thing is if the patient has a mutation in CD40, so you can't have this interaction, never get class switch, never have germinal centers, always make IgM forever. The other protein is CD40 ligand. If you have a mutation in CD40 ligand, the T cell can't provide the signal to, you don't get any of this. The third protein I'll mention in a second. Right, so that's class switch. It's hyper IgM. So here is our general outcome of the germinal center reaction. We get high-level antibody secretion, class switch, affinity maturation. Potentially, a B cell can go to becoming a memory cell, or it can go to becoming a plasma cell. And this is actually a plasma cell under a microscope. And so you can see it just turns into basically a cell full of ER, ready to secrete stuff. And so we will have a whole lot of plasma cells made, though there will also be some memory cells that are made. And so you can see some big differences. In fact, the plasma cells make antibody, and they only make antibody. That's all they do, high-level antibody all the time. They stop having B cell receptor on their surface. They stop having class 2 on their surface. They don't grow anymore. They don't mutate anymore. They don't switch anymore. They do nothing except make high levels of antibodies. I felt really bad earlier this semester because over the summer, I got this fabulous t-shirt about cells of the immune system, and it has different kinds of cells in the immune system, and as they're little people, and they say different things. I love that shirt. It's a great shirt. I was going to wear it when you did the microscopy lab, and I totally forgot. I'll wear it someday for you. But the reason why I'm mentioning it is because it has plasma cell on it. And some people, some immunologists I know, we like had a debate about why plasma cell was saying what it was saying. Because plasma cell on that shirt says, I don't care. And my answer is, because plasma cells have no receptors and do absolutely nothing and pay attention to nothing other than making antibodies. Plasma cells just make antibodies. They don't care about anything else. So that's why my plasma cell on my t-shirt says, I don't care. So this information is what we knew about all of this when I finished undergrad. However, in the time since then, we actually learned, and when we did that, so we used to say, and then hypermutation happens, and then class switch happens. And while I was in graduate school, somebody actually found an enzyme. And the thing that's cool about this enzyme is that this enzyme actually is responsible for both class switch and somatic hypermutation. So even though they seem like two totally different processes, they actually both go through the same enzyme. So the cell only has to turn on the one enzyme, and it can do both of these things.